Hello everyone, welcome back to my channel. For those of you who are new, my name is Catherine and I hand draw science videos, especially about emerging therapeutics and diagnostics. Today, we're going to explore something called organs on a chip and its potential to replace animal testing in drug development. First of all, let me give you a brief overview of how a medicine is developed. First, there's this drug discovery stage, and after that, a potential drug candidate is identified. Then this candidate would go through preclinical studies in vitro using cells and in vivo using animal models. If the candidate shows safety and efficacy in animals, then it can be tested in humans during clinical trials. Finally, if there is enough evidence from these trials showing positive results, the drug can gain regulatory approval to be brought into clinic. As you can imagine, the problem is that this is very time-consuming and expensive. In fact, it was estimated that it can take 15 years and cost up to $2.6 billion to make one medicine. Also, more than 80% of the drug candidates entering clinical trials fail to be approved in the end, which is a big waste of both money and time. Now, one of the major factors contributing to this is the animal testing stage. It can take years to complete and often do not predict the correct response in humans because many human diseases don't have an animal equivalent. Considering the economical and ethical issues with animal testing, scientists are now looking for better ways of doing this, and one potential solution is organs on a chip. So what are organs on a chip and how can they help? Organs on a chip are made up of real living human cells that mimic the functions and structures of a real 3D organ system. The first lung on a chip research was published in 2010. It's around the size of a memory stick and is made up of a clear block of polymer which has two tiny channels separated by a thin membrane. In one channel, it's lined with human lung cells, and this is where air flows through. Meanwhile, the other channel is lined with blood vessel cells. This is where a nutrient-rich liquid flows through, mimicking the blood flow. There is also a vacuum applied to the lung chip to recreate the breathing movements of the lungs. So you can see that although the chip looks nothing like the lungs, the structure and the different cell types present do resemble the lungs. Over the past decade, scientists have developed different chips mimicking organs like the heart, kidneys, liver, intestines, and bone marrow. Different organ chip platforms have different designs and sizes, but essentially they have three things in common: having a 3D arrangement, having multiple cell types to reflect a more physiological balance of cells, and finally, using mechanical forces relevant to the tissue being modeled. So, what are the advantages of organs on a chip? Firstly, scientists can have precise control over the cellular and specific tissue architecture. They can also incorporate real-time tissue function sensors to better monitor cell activity. Secondly, this technology can potentially be applied in precision medicine, which is an approach to tailor effective treatment to specific subgroups of patients by making patients on a chip. Imagine if we make organ chips from patients that represent certain genetic subpopulations, and we use these chips to test whether a certain drug works. Then, we can use the same cohort of patients for a more focused human clinical trial. This would increase the likelihood of success, shorten the time required, and reduce cost of developing the medicine. Thirdly, it might even be possible to apply organs on a chip to a personalized medicine approach using cells or tissues derived from individuals to test which specific drug will help each patient best. Moving forward, scientists are now working on body-on-chips made up of multiple human organ chips as an integrated system. They do this by using channels lined with blood vessel cells to transfer fluids between the different chips. The goal here is to model complex organ-organ interactions and uncover the pharmacokinetics and pharmacodynamic information. By modeling different organs, it can also allow us to better understand the off-target effects of a certain drug. Let's take a look at this example, which is a chip developed by the DARPA. As you can see, it interlinks 10 different organ systems. To validate the platform, researchers are now testing compounds with known effects in humans on the chip to see how good it is at predicting the drug effect. So moving forward, Can organs on a chip actually replace animal testing? If animal testing were to be replaced, this is essentially revolutionizing the whole regulatory process and the pharmaceutical industry. One of the biggest challenges would be proving that organs on a chip are as robust as animal models and that they can produce more meaningful results. So it isn't likely that we can achieve this in the short term. However, even if animal testing can't be replaced completely, organs on a chip would be a great additional tool to help reduce the current reliance on animal models. And even just speeding up the process of drug development and making it cheaper would make a huge difference in terms of getting medicines to the clinic and eventually allowing better patient access to treatment. So that's the end of my video today. If you enjoyed this, make sure you comment below and subscribe to my channel. Follow me on Instagram as well to stay tuned, and I'll see you in my next video.